Good morning, this is Nasira Bellawar, fourth year in Applied Chemistry at Google University. My abstract is about a comparative study between the insecticidal potential of Calotropis procera latex against Parlatoria blanchardi and commercial insecticides under the supervision of Mbarka Bouziane, Lakhdar Hamdi Isa, and Haj Mohammed Mahfoud. In North Africa, the date palm constitutes the main living resource for the inhabitants of desert areas. It plays an important role at the social and economic level. In Algeria, date palms occupy about 167 hectares, with 80.5 million palm trees producing nearly about 1 million tons of date per year. Unfortunately, Parlatoria blanchardi is a threat to this natural resource. Indeed, it is among the main pest species threatening date palm cultivation in this region. The white cochineal considered one of the most formidable pests for a long time in the Algerian oases. Indeed, the use of pesticides boosts crop productivity and improved product quality, but there are no concerns about overuse and consequent negative effects both on wildlife and human health. For this purpose, scientists resorted to searching for a natural alternative to these harmful pesticides. The present study was conducted to determine the phytochemical composition of Calotropis procera latex. It also consists in evaluating the latex insecticidal effect compared to the commercial insecticides. Calotropis procera is one of the most famous plants that humans have used since ancient times in the treatment of many human or animal diseases. Previous investigations on Calotropis procera have been shown to contain insecticidal properties. About material and methods, the samples of Calotropis procera latex are collected from the region of Janat at Tassili, Niger. Regarding the insects, Parlatoria blanchardi individuals were collected from the middle leaf of the Glycnor date palm, cultivated in one of the oases of Ghardaya province. In order to inspect the insecticidal activity against Parlatoria blanchardi, different aqueous concentrations of latex and commercial products were prepared. A sample of crude Calotropis procera latex was analyzed qualitatively to determine the presence of secondary metabolites. As it is mentioned in the table, the phytochemical tests revealed the richness of Calotropis procera latex in secondary metabolites. It contains alkaloids, phenols, terpenoids, steroids, glycosides, and large quantity of cardiac glycosides was detected. On the other hand, the flavonoids, tannins, saponins were not detected. Several previous reports indicated that the plant contains various classes of secondary metabolites, including proteolytic enzymes, carotenoids, alkaloids, tannins, triterpenes, and flavonoids. As it is now, all of these natural products exhibited diverse biological activity such as cytotoxic, anticancerous, anti-inflammatory, and analgesics. After counting the number of living individuals, male, female, first and second fixed larvae, 3 pieces of leaflets were put in glass petri dishes that were containing soaked cotton with distilled water. After that, the aqueous solutions of the latex as well as the solutions of the commercial insecticide as positive controls were sprayed. Each treatment was replicated 3 times and the incubation was at 13 degrees Celsius, lasted 24 hours. Tried the insecticidal rate shown on Fig 1. It could be noted that the mortality rate of Parlatoria blanchardi started progressively from 0.009 up to 15 microliter per milliliter of tested samples. From 0.075 to 15 microliter per milliliter, their action was too close, but at 15 and also at 7.5 microliter per milliliter, the latex and the commercial insecticide showed a very similar toxicity on the insects. On the other hand, at lower concentration 0.037, 0.018 and 0.009 microliter per milliliter, Calotropis procera latex exhibited the highest rate with a percentage of 49, 38 and 24% respectively, compared to the commercial insecticide with a rate of 32, 17 and 12% respectively also. These interesting results confirmed the efficiency of Calotropis procera latex on Parlatoria blanchardi larvae and adults compared to the commercial insecticides. Referring to LD50 results, it is clear that the latex sample is potentially very toxic on all stages of Parlatoria blanchardi, in particular on the first and the second larva. The corresponding LD50 were 0.1 and 0.057 microliter per milliliter respectively, while the LD50 values on male and female insects were 0.75 and 0.54 microliter per milliliter respectively. Furthermore, The study realized carried out by Marcock showed that C. procera latex caused 50% mortality against Alabranchia. Similarly, Shai found that latex is a strong killer against both of C. ex cuncta fasciitis and I. N. Stephen C. larvae compared to the other Calotropis procera extract. Another study showed that C. procera latex contained the larvicidal compound, which after 5 minutes caused 100% mortality in the third stage larva of Aedes aegypti. Besides, the commercial insecticide showed remarkable results, but its action on female was the most important, with an LD15 of 0.12 microliter per milliliter. As a conclusion, the crude latex produced by the green parts of Calotropis procera was evaluated for its toxicity against Parlatoria blanchardi, adults and larvae stages. The results showed that the latex possesses insecticidal and larvicidal activities on Parlatoria blanchardi, even more than commercial insecticides. Other studies must be carried out to use this latex as a suitable alternative product for pest control and to avoid chemical damage to trees due by synthetic insecticides. Thank you for your attention.